Welcome to the day 7 session on sequence analysis, PAM, BLOSSUM, BLAST, types of BLAST, and how the BLAST algorithm works. So we'll be discussing about these concepts in day 7 in sequence analysis. And if you see the content, so according to the training schedule, we'll be we are in the day 7, so we are completing this. And tomorrow we have session on FASTA, PHA blast, and PSA blast. Okay, in sequence analysis, and also we'll be discussing about motive finding, motive elicitation. So all these things, and we can see even other tools in sequence alignment other than this. Okay, so we discuss other tools in sequence analysis on tomorrow, if possible, because in phylogeny we have a lot of basics, so I can also cover that in the tomorrow session. Okay, now let me complete the today's session. Before going to these topics, PAM, BLOSSUM, BLAST, and BLAST algorithm, can we recap the last session contents? So we can recap the last session topics. Participants join on time. You know, at 7 o'clock, only 15 participants are joined. So try to join on time. So you may be missing some session. Okay. Okay, let me discuss about the last session topics. So interact participants, then only even I can show interest on you on discussion of more concepts. Very good. We have discussed something related to Needleman-Wundt algorithm. So that is, can someone tell me what is the alignment used by Needleman-Wundt algorithm? The type of alignment used by alignment, I mean type of sequence alignment. Sequence alignment also called as pattern matching. Okay, very good. The answer is global alignment. And obviously Smith-Waterman uses, Smith-Waterman going to use local alignment. Good. So this is what we have discussed. We have seen the problems, how to use the matrix method for Needleman-Wundt and Smith-Waterman, which comes under dynamic programming. How to go for calculation of alignment score. You remember we have done two one is calculation of alignment score. The second task is to go for finding the best sequence alignment. Finding the best sequence alignment. These are the two outputs we have done on yesterday. Okay, we have solved a matrix that we put a query in a, row wise and a database in the column wise. We can also alter. Okay, instead of row wise, you can also put query into column wise. If you put in column wise, obviously the database sequence you should put in row wise. And you can either use up down approach, okay, or you can use down up approach. Okay, so whatever you feel best, you can use it, and whatever you feel comfortable, we can try it out. But the algorithm steps is common, isn't it? So still we haven't received the recorded session yesterday. Okay, I just follow your coordinator. I mean, follow your coordinator and ask him to send. Okay, don't worry. Actually, today you'll be getting if you didn't receive it on yesterday. Okay, so today you'll be getting that. Initialization, iteration, termination. Very good. So before asking the question, you are alert that I will be asking this question. So the three steps in both the Needleman-Wundt and Smith-Waterman algorithm is initialization, iteration, and termination. You are right. What are the what is the difference between Needleman-Wundt and Smith-Waterman algorithm? Whether all these three steps remain same, what is similar and what is different? Now I ask you, what is similar in Needleman-Wundt and Smith-Waterman algorithm? I am asking about the steps. Which step is similar? You have mentioned three steps. Good. Initialization, iteration, termination is the correct answer. But what step is same in both the algorithms? Initialization? No. No, I think you are studying BTech, but very good. I appreciate Pooja and Kirtana. Okay, they are trying from the beginning. How about the others? Even others can try. During initialization, we put zero. Very good. You are trying once again, Shreyasi. You are right. But whatever we put zero for the first row and first column is only for Smith-Waterman. In Needleman-Wundt algorithm, after first cell, if you proceed to the next cell, box beside or box up, you add the gap value like -2, -4, -6. You remember on yesterday for Needleman-Wundt for the initialization step, you find Needleman-Wundt and Smith-Waterman vary. For Smith-Waterman, the first row and first column you put as zeros. But Needleman-Wundt, not like that. 0, -2, -4. You remember? That is initialization step. So obviously can find initialization in Needleman-Wundt and Smith-Waterman is varying. Okay. Then what is same? If not initialization, what step is same? So, okay, I understand you didn't receive the recorded video. Good. But I think you are struggling because of that. I can show you with an example. Okay, don't worry, participants. I understand. Okay, but if you receive the video, you can also watch that. Okay, so definitely may have an idea. Okay, so if you see this Needleman-Wundt algorithm, okay, so we just have a recap on what we discussed on yesterday. You see the initialization, iteration, and termination. Initialization, the first cell alone is 0, comma 0 is 0, and if it is 0th row, first column. If you take, because we will be start from first column and then go for second column, then third column. So the J alone will be getting varied. But the initialization is generally in the first row and first column. So this is only one, and this is also only one. Okay, so the next column you get it means, and it's not into actually you are sum up with the gap value. This is a sum up of gap value. Okay, so this is the initialization step. We follow the Needleman ones, and iteration. You remember how we have calculated. So in initialization step is different. Here you find -2, -4, -6, -2, -4, -6, -8. But whereas here also you can see, if you see the Wikipedia, you get a good kind of an idea because here you find, you remember on yesterday, whatever we have gone for sequence alignment, we got only one based alignment. But if you take this large sequence, if you take this much large sequence in Quarian database, you got nearly 4 different based alignments. Not a single alignment, you got 4 based alignments. Okay, and even you get the scores like that, or you get 4 different scores. Okay, one should know even how the 4 different alignments came in Wikipedia. Okay, in Wikipedia, in Needleman ones, they have given this kind of a table, and they have also given this kind of an calculations. 4 different alignments are shown because they have used the up bottom approach. Okay, I started from bottom to up, but here they have used like up down approach. If you see the up down approach while doing the back tracing, okay, from here to here is okay. You get one path like this, and you are also getting second path like this. Like how in Google Maps you get different path to move. Okay, but we choose which is the best, which is the shortest, or which path shows you the less time to travel, isn't it? Similarly, here if you want to go for calculation of alignment score, okay, here you have one path is like this, going directly like this, and you'll be joining like this. I can use the pen, isn't it? So I can use a pen. 1 minute. Okay, it's in the red color. So one path is like this, travel like this. Even it can travel like this. And now it joins like this. One path is like this. Now I can go for the second path. See the second path. The second path again, it goes like this. The second path is going like this. But here you can find the deviation. So second path is going like this now, and it joins here. And there is also third path. You know the third path. See the third path again. It joins here. It comes like this. Third path, but it goes like this, and the third path will be transverse like this, and it merges with this path. So there are the three different paths we found, isn't it? And the fourth path is something after this we find like this. Okay, actually, majorly I found the three alignments. The fourth alignment is this. This is already found. The fourth alignment we follow like this. We follow like this. I found majorly three best alignments. Okay, the red, green, and blue. Okay, I think this is the raw sequence, initial sequence what you take, and after taking this, okay, yeah, this is right. You get only three sequences. These are the inputs. These are the inputs. Actually, this they'll be providing the data. So the best alignments so one and best alignment two. This is best alignment three. So whatever I showed in three different colors or three different alignments. This is taken from Wikipedia. So one can search the Needleman ones and it can come to know how the scores are calculated, how the obviously the best alignment score is the last cell value. So this is the termination step. In the termination step, whatever value you find at the end is zero. Okay, I mean, you got a zero. So this is the alignment score. You don't have variation in scores. Only one score you have in the Needleman ones. For this problem, we got alignment score equal zero. No doubt in that. But you also have find different paths. You also find different paths. Okay. And you got for the different path, for each and every path, you got a kind of a sequence alignment like this. So in between, you're introducing the gaps. Okay. So just watch through that. And if you have any doubts, you can also discuss this in the next session. Now, like this, okay. Neilman, once we can go to the next. System got hanged. Okay. The Smith Waterman is not like the previous one. So instead of using f of 0, here you can see all the three cells are 0. So f of 0 comma 0 is also 0. F of 0 comma j is also 0. I is also 0. But previous case in element ones, you have f of 0 comma j equals minus d. Whatever value you have, minus d, isn't it? But here you don't have that distance value. You're not going to add the gap value. Okay, so this is the difference. So initialization, you find something different, and also termination is different because in termination we take only no, we are not taking the last value, but we take the value which is maximum of all the cells. Okay, if you see this, this is the maximum value I found. So the alignment score for this problem, what we solved on yesterday, is 2, isn't it? We are not taking in the last cell, but we are taking in the we are comparing all the cells and we found that this is the maximum value. So the termination is also different. So only thing is common is only the iteration step. The iteration step alone is common. Whatever you calculate, three value, all these things are common only in Smith Waterman and Needleman ones. The remaining two initialization and termination steps are different. Okay, so this is extracted from Wikipedia. Okay, if you search for the Smith Waterman, you get like this. Okay, and this is a large sequence, of course, but you get a kind of a path. Okay, they just started from here. Okay, so they started from bottom, I mean, so top, and they ended up to the bottom. But you got the maximum value only here. Okay, where you got the maximum value, you got this as the maximum value out of any other cell values. So obviously, this is your score, but the paths are different. One path is like this, and another path. If you have different paths, you have so you can show the different alignments. One score, but different alignments. Okay. So the alignments is based on how you got, how you find a trace back. Okay. Okay, now we will be discussing something related to motive generation. What is motive and how to generate the motives, and how to generate a single motive and how to generate the multiple motive. Motive is generally a part of a protein which gives you the function or a structure. There are types of proteins like structural proteins, functional proteins, isn't it? So if you go for structural proteins, whatever important part of a protein which gives you a structure, that we call it as a structural motif. The best example I can provide is the myoglobin. Myoglobin is a muscle protein. In this muscle protein, which gives the strength, okay, so which gives the structure. So that part alone, we call that as a structural motif of a myoglobin. But whereas if you see the insulin protein, another example, the insulin is not a structural protein, but that is a functional protein. This protein is majorly used to regulate the glucose level. So the main function of insulin is to regulate the glucose level. So in that case, we consider that motif as a functional motif. Okay, we can also have another type of classification like a comparative kind of a motif, where we can go for structural or functional kind of a motifs. Now let me take you to the software tools on how to find the motif. For this, first I can use Scan Pro site. Okay, so this is the tool we use for searching a single motif. Okay, this is present in Xpassi website. In Xpassi website, we have a program called Scan Pro site. In the Scan Pro site, you have to paste the FASTA. Okay, so for getting the FASTA, better go to the Uniprot website. In Uniprot website, okay, take a protein. Okay, so from the beginning, we are taking the human insulin, isn't it? So we take that insulin and we search for the functional part of a protein. Okay. If that is the task, okay. So type insulin. So in insulin, we have generally insulin ligand and insulin receptor. So if you want to find from a small protein, that is a insulin ligand, okay, fetch the FASTA format. Okay. So for this, I just get inside and I can go with the download. Okay. So I want data setter. No, I don't want the FASTA. So if you want faster format, I better go for sequence and isoform. Okay, in sequence and isoform, I got like this. Okay, 110 amino acid, insulin is here. Just click download. If you click download, you get the sequence. Just copy the sequence. Now, I have to either provide the Uniprot ID. So if you already have that six-letter ID, you can paste it. P01308. Okay, so that is the identifier. Either you can give like that, or even you can give the sequence in FASTA format. Anything it accepts. So again, I'm repeating. This is the tool used for finding the motif. In our case, we have taken the human insulin to find the functional motif of insulin. So I fetched a protein sequence, and then you can able to start. If you want to receive it to the mail, you can also check this. But you get results as soon as possible. No need to check that and provide the email. Okay, simply click start the scan. So now for the protein, you get a kind of a functional motif. For 110 amino acid, if you see here, so I'm just keeping my pointer here, you can able to find 95 amino acid to 109 amino acid. 95 to the 109 amino acid is the functional protein, functional motif of this insulin protein. Okay, so this is what we got. And if you want to find the same for a receptor, we also have some other legends also. Okay, so if it is, so it actually comes under a kind of an a binding site. Okay, if it is an enzyme, we call that as active site. It's not showing disulfide bridges, active site, other ranges, other sites. But whatever the orange color here was shown is the functional part of a protein, insulin protein. Now, instead of insulin ligand, can we drive the insulin receptor? Can we check what exactly we'll be getting for that? Simply go to the entry, go to the sequence, and click download. The 1382 is the length of the insulin receptor protein. Select it. Instead of the previous ligand, I paste the receptor, or you just give the Uniprot ID. Click start the scan. So based on the length of a protein, it takes time. But okay, we got this. So see this. There are three functional parts of a protein. Okay, one is fibronectin type 3 domain. This is fibronectin domain. This is also fibronectin domain. And this is protein kinase domain. So more about this you can see in bottom. Yeah. So you got this. So more about this, you got the information in the bottom. So the three motif, whatever you find, cumulatively they are showing here. And if you click this, okay, you also get this information. What is protein kinase? And if you get the references, description, technical section. So you can also click this, so it take you to the hyperlinks. So you can find this diamond symbol. Diamond symbol is what? Active site. So you find one active site for the receptor. What do you mean by active site? Actually, the insulin ligand generally binds with the insulin receptor in this position. Okay, you know, in immunology, the immune ligand proteins bind with the corresponding immune receptor proteins. In our case, insulin ligand is going to bind with the insulin receptor. If so, this is the binding site. Okay, that is the binding site is 1155. I got earlier. Take some time. Okay, 1155 to so that is the position. Okay, if you keep here 1155 to 1167, this is what we call it as protein kinase domain. The protein kinase domain is what? The binding site. Okay, so this is something about scan pro site, which is present in XPASI Web2. Similarly, instead of focusing on a single motif, if you want to go for multiple motifs, I mean, if you have a multiple proteins, nearly 3 to 4 proteins, and if you want to get what are the, because this is what we used at local and globally, isn't it? If you want to, if you have a similar kind of conserved regions, okay, if you want to get a maximum alignment score, okay, I ask you to go for local alignment instead of global. You remember? So similarly, if you go for, if you type MEME, you get so many memes. Don't go to that. ME also type this MEME followed by suit. So this is actually multiple illustration motif excitation suit. Okay, actually I can show you. Uh, yeah, multiple EM for motif illustration. Okay, MEME is multiple EM for motif illustration. There are so many functions. This is a kind of a pipeline how we can use and what are the different tools we can use in this MEME suite. MEME suite is a package. We have so many tools to work on sequence alignment for motif finding, something like that. So for each and everything they have shown is here. Motif discovery, motif enrichment, gene ontology databases, discovery motif. So for each and everything, you have some collection of tools which you can use it. Now presently, if you want to, if you have a 3 or 4 proteins and you want to find motif for all the 3 or 4 proteins, then you can click here, MEM. You have this icon, click this. Now it's asking for to provide an input of FASTA format. Input the primary sequences. You have an option for uploading the FASTA format. Don't choose this, but choose this. You have a choose file here, input the primary sequence, you have to input that. So for this, you should have a FASTA format with 3 or 4 sequences. I think I already teach you how to download 3 or 4 sequences in a single FASTA format. How can we make it? Do Uniprot, isn't it? You remember? So last time I told you if you want to compare, okay, human insulin ligand, zebrafish, ox, China pig, okay, and if you want to have pig also, okay. So I just selected 5 sequences. If you want to download this, okay, click download. You can download in no compressed format, FASTA canonical. Okay. Even last time you have downloaded like this. So no need to go for manual kind of inputs. Okay, simply just like that, you select it and it can download as such. In desktop, we have bioinformatics demo. We have insulin 3 proteins. Now we have insulin 5 proteins, isn't it? So I give 5 proteins. So default it's in FASTA file. Okay. Click save. One thing you have to remember is don't download in compressed format. If you compress it, then you have to uncompress. So I remove the compression and just download the FASTA format. Now it's already available in desktop. Simply upload it. Further, click choose file. Click choose file. In desktop, I have a folder called bioinformatics demo where I have insulin 5 proteins. Click open. Now I input the primary sequences. So the email address, if it, you can give, okay, and a job description. It is actually insulin 5 proteins motif finding, isn't it? So some kind of an uh, text, okay, the file name or a job name for the output. It takes less time only. It won't take much time, but still, for my safety purpose, I've given like that. Okay, even the job will be a link will be transferred. Okay, after the job is generated from this tool. So submitted. It takes some time. I didn't get it. If I receive it, we can see that. Okay, take some time, wait for some time because I just not tried with a single or 2 or 3 sequences. I tried with 5 sequences. It takes some time accordingly. Okay, still it's running. Now let me go to the next chapter, that is PAM and BLOSSUM. So today we'll be discussing on these topics, PAM and BLOSSUM. So if you see PAM, okay, the PAM, BLOSSUM, etc. This comes under amino acid scoring substitution matrices or amino acid substitution scoring matrices. Okay. So what do you mean by that? Why we have to introduce all this? First, we understand what is PAM and BLOSSUM. Then I can tell you the exact application. Why we have to use this. PAM, generally we call that as a percent accepted mutation. You know, the father of bioinformatics, I told you, is a former director of NCBA. I also told you it's not a father, actually it's a mother. Margaret Dayoff. Margaret Dayoff is the discoverer of PAM. I think this discovered in 1970s. Okay, so she has tried everything through the calculations and we found the mutation rates. And of course, during that time, only even the NCBI came into play. I mean, NCBI came to existence. Okay, likewise, BLOSSUM. These are other amino acid substitution scoring matrices. Okay, the BLOSSUM is the other, and the expansion is blocks substitution matrix. Then what is the difference between in the PAM and BLOSSUM? We also have so many scoring matrices. We have GONET, we have other matrices also. But that role is rarely in use. Most of the public domain databases uses these amino acid scoring matrices. Okay, that is the databases with pairwise sequence alignment tools. We have tools like BLAST, FASTA, etc., isn't it? So these are the tools we generally use it for pairwise sequence alignment. For going with these tools, okay, we have these options to select. One can also have these additional options to select. Then what are these options? Why these are used? We see in the subsequent slides. Before that, we go with the PAM in detail. The PAM I already told percent accepted mutation. So what is it actually? This is actually, yeah, it's the definition is mentioned in the next slide. That is, one PAM is generally 1 point mutation per 100 amino acids. If you have a protein with 110 amino acid, okay, even if you have, that doesn't mean the protein with 110 amino acid or even a protein with 1333 amino acid like glycoprotein, whatever proteins you have, okay, this is the common concept we adopt for all the proteins. We just select the 100 amino acids initially. Even if it is 110, only 100 amino acids are you take into account because you don't have the another 100 amino acids. You have only 110 is close to 100. And if you take 1333, you are almost you have 13 amino acids comes into play, isn't it? So here, what they mean is, for 100 amino acids, if you have 1 point mutation, then it is 1 PAM. In 1333 amino acids, if you have 13 point mutations for the 1330 amino acids, then that is 1 PAM. Okay. So for 100 amino acids, 1 point mutation. And if that is PAM, what is BLOSSUM? The BLOSSUM is discovered by Henikoff. You know, okay, according to BLOSSUM, the BLOSSUM 100 is derived from sequence block clustered at 100% identity level. Do you understand something? So actually here BLOSSUM, okay, if you have BLOSSUM 100, this is the in BLOSSUM we have range from BLOSSUM, okay, we have range from BLOSSUM 10 to 100. This is the range for BLOSSUM. If you have BLOSSUM 100, okay, that's a value, okay, in this case. It means that if you compare sequence 1 and sequence 2, both the sequences have 100% similarities. If both the sequences are similar, it might be a close ancestral species. For example, most of the proteins of human match with chimpanzees. Most of the proteins match human with the chimpanzee. So, in that case, for the proteins, you can use BLOSSUM 100. Okay, but if you see the range is 10 to 100. You don't have 1 to 9. If that is the case, how about PAM? For a PAM, okay, we have values from 1 to 250. This is the range. PAM, use the values from 1 to 250. Okay, whereas BLOSSUM is 10 to 100. You have to remember that BLOSSUM value gives you the percentage similarities in the sequence. But PAM, we represent PAM1 represent 1 point mutation per 100 amino acid. If PAM 250 is mentioned, then 250 point mutations per 100 amino acids. The mutation rate is more. When we can go for mutation rate more, this is taken in special case like SARS-CoV-2 virus. In SARS-CoV-2, you might have heard there are so many kind of strains. Initial strain, from the initial strain, you can find alpha, beta, gamma, delta. So many strains are there, and each and every strain has some 7 to 8 mutations in average. So totally, you can get almost nearer to 100 kind of mutations in SARS-CoV-2 viruses. Wherever you find mutations are more in the species, then we can better go for PAM. Okay, and if you see, there are few species where you find won't mutations. Okay, there you can find the PAM value is very less. If PAM value is more, you can for this I can take an example like SARS-CoV-2. But if you see the species where you don't find any mutations, for example, Nipah virus you have only one strain. So for Nipah virus, you can use this PAM1 instead of PAM 250. Okay. So now even for further, if you want to know more about the PAM and BLOSSUM, we also given some examples. Like if you compare PAM with the BLOSSUM, how can we compare? So this we can see with an example. PAM, that is which we call it as a percent accepted mutation, is more frequently used for sequences which is closely related. I told you most of the proteins of chimpanzees and human are closely related to each other. Okay. So in this case, you should not go for BLOSSUM. Instead of BLOSSUM, I ask you to choose PAM. Okay. The reason behind we see in the next slides. So, but this is the exact example. Okay. So for close related species, go for PAM. If you find distant related species, E. coli, comparing E. coli with human. Is this possible in practice? Why we have to compare E. coli with human? This is the question now. So the participants can try now. So chimpanzee, human, we compare to just check the relation. Okay, hereditary and other kind of mutations, everything we check from here. But why we have to compare E. coli and human? So this is an example they provided for BLOSSUM. Can someone tell me why we have to compare E. coli and BLOSSUM? Yeah, dear Krishna, actually, motive discovery, enrichment. So all these things we use, okay, when you go for checking the conserved regions. Okay, and so this is generally used based on the project. Okay, when you need motives, and for example, if you take even SARS-CoV-2, alpha, beta, gamma, delta. Okay, you have only one or two amino acids are mutated, and but you can't tell the structures from the structures that ACE is going to bind with the same binding site of that mutated SARS-CoV-2 spike glycoprotein. It depends. So the motives are most important. Okay. So in motive, not only we have sequence motive and functional motive, we also have structural motive. The structural motive is dependent on the structural protein. If you take a structure of a protein in a spike glycoprotein of a normal virus, and if you see the mutated virus spike glycoprotein, you find the variation in structure, especially in binding site domain. The binding site domain varies. So one should also know. Okay, so in this COVID-19 infection itself, one should know all this. And if you are comparing with other species, obviously one should go for this kind of an enrichment scanning comparison, etc. Okay, I just ask you a question. Why we have to compare human with E. coli? Any reasons, specific reasons? I told you that human and chimpanzee can be compared. Okay, most of the cases, and we can use PAM. Then when can we use BLOSSUM to study evolutionary relationship? The evolutionary relationship is what, PAM, closely related species, human and chimpanzee. But whether E. coli, the humans are evolved from the E. coli. Do you mean that? I told you for blossom, yeah, I understand. But why we are using blossom like this? More distant related species. I give an example, human E. coli. You didn't, others don't get. No, so if you don't get, I can provide you the answer now. So the reason behind is most of the proteins in human, okay, are genetically engineered. You take an example of insulin. How we are getting this recombinant insulin, fast insulin, pro insulin, so many insulins we are getting, isn't it, in market? Earlier, we have to sacrifice millions of billions of pigs for getting the insulin. But now we are going for genetic engineering. We are using E. coli and we are going for genetic engineering and we are producing the recombinant insulin. So, if you want to incorporate the human gene in the E. coli plasmid to construct a vector, obviously one should know even the similarities of the E. coli plasmids and the human incorporating insert gene, isn't it? So one should also have a knowledge on these comparisons. So for these kind of a distant related species like E. coli and human, in our case, we are taken to produce a recombinant insulin, we can go for blossom. But you can't go like this blossom 100, but you can go with blossom 10. In blossom 10, it means that you have only 10% similarities. Even if you have 10% similarities in E. coli and human, it may give a kind of a better result during the genetic engineering technique. Blossom 10 is just 10% similarities in sequence 1 and sequence 2 of a particular protein. But Blossom 100, again, if you take this number, this can be used for comparing chimpanzee and human. But why we can't use chimpanzee and human comparison? I mean, why can't we use this kind of an E. coli and human comparison in PAM? Why can't we use? So that we'll be seeing in the next slides. Okay. So before this, so you should also understand some information why we have to use the amino acid substitution scoring matrix. Here they have given an example and how to use it. You know, I told you PAM is 1 PAM equals 1 point mutation per 100 amino acids. And if you substitute a D amino acid, what is D actually? Singulated D amino acid. Unmute and answer. What is D and what is E? Can someone unmute and answer? What are D and E singulated amino acids? No, your headphone is not working. You can type the answers in the chat box. What is a singulated D amino acid and what is singulated? And also, so participants, if you're not understanding, kindly ask. I don't know, you're maintaining silence. I'm scared. Any doubts you have at present? Very good. Answers I got from Anand Kumar. Aspartic acid and glutamic acid is exact. Okay. So that are the exact answers. Actually, aspartic acid molecular weight is less and even acidic value is comparatively less. The aspartic acid acidic value is less when compared to glutamic acid, isn't it? Even though both are acidic amino acids. So if you find a sense mutation, there are the types of mutation, isn't it? Sense mutation, missense mutation, nonsense mutation. Even missense mutation is bearable. Nonsense mutation is not accepted. The best example I can provide for nonsense. This is a sense mutation. Okay, so the acidity is increased. If D is substituted with E, you can add 3 to the score. You can add 3. Where you can add 3 to the score? You can add 3 in that matrix table in Needleman-Wunsch or Smith-Waterman. If you're using the global alignment, okay, basically it is using the global alignment, of course. If you use a Needleman-Wunsch algorithm, and I tell you an example. So you remember in Needleman-Wunsch algorithm, I told you like how to go for calculation of how to go for iterative step. Okay, if you have an amino acid sequence in the rows and columns, if you have a D, instead of D, if you have E, okay, here, if you have a 0, I told you to add +3. So instead of 0, okay, if you add +3, this is an additional thing used. Of course, this value got from 3 values. Box beside, you got from this value, and one value you got from box here, and another value got from the diagonal. From the 3 values, you got the 0. The optimized value is 0. But whatever optimized value 0 you got, if you use amino acid substitution scoring matrix like PAM, okay, and if you find D instead of D and E amino acid is substituted here, then you get a score like 3. And whatever score 3 you use it for further calculations. This is how we make it out. Okay, there's an additional thing we incorporate in the matrix. Okay. So only we call this as amino acid substitution scoring matrices. So instead of D substituting with E, that is which we call that as a sense mutation. Okay, this is a sense mutation. Instead of D, if you find this is substituted with K, what is K? K is lysine. How about the nature of amino acid? This is a basic amino acid. This is not an acidic, basic. If you are mutating D with K, then no, not this. Substitution of the positively charged K for the non-polar proline. Non-polar proline. This is a polar and this is non-polar. Okay, and even if you're substituting instead of acidic, if you're substituting with a basic amino acid, you can give a negative score. Even that is one example. This is another example. Okay, even if you substitute K with a P, you have to give a negative value instead of positive value. In our case, we have given like -1. So instead of 0, if you find you have to add like -1 there. So which we consider as nonsense mutation. Missense, kindly ignore. No values are substituted. No need to substitute 3, no need to substitute -1, simply +0. So ignore that. That is missense mutation. So if you see this kind of a difference between PAM and BLOSSUM, this is the important slide. How when we have to select PAM, when we have to select BLOSSUM. So if you see this slide, you can able to understand. Already to mentioned you, we have PAM range from PAM 1 to PAM 250. So the PAM 1 to PAM 250 range is spanning only BLOSSUM 45 to BLOSSUM 80. I told you in BLOSSUM you have range up to BLOSSUM 100, but this is not covered in PAM. This is not covered in PAM. There is no negative value in PAM. Only have value from PAM 1 to PAM 250. How about the least value in BLOSSUM? BLOSSUM, we have least value up to 10. We don't have any other PAM values here also. So BLOSSUM 100 to 10 is a maximum span range when compared to PAM 1 to PAM 250. So when we go for this maximum BLOSSUM 10, if you find more dissimilarities in sequences, if you have sequence 1 and sequence 2, in our example, I told you take the human and just compare with the E. coli. Okay, where you find more dissimilarities. For more dissimilarities, that is 45 to 10. You understand? You can use BLOSSUM. If you have dissimilar species, okay, not like ancestral species, better go for BLOSSUM. If you have more similarities because the range is very less, okay, we can go for PAM. That is comparing human and chimpanzee. Okay, and you can also find which is less divergent in common. Okay, if PAM 1 is equivalent is BLOSSUM 80 and PAM 250 equivalent is BLOSSUM 45 and PAM 120 equivalent is BLOSSUM 62. The default amino acid substitution scoring matrix used in BLAST is BLOSSUM 62. But we also have BLOSSUM 45. If you want to check with PAM 250, you can also select BLOSSUM 45. Okay, even we have BLOSSUM 80. If you want to check with PAM 1, okay, very less amino acid substitution, then you can check BLOSSUM 80. You have these three values in BLAST. One can select this, but default thing what we use in BLAST is BLOSSUM 62, which uses PAM 120. That is 120 single point mutation, and amino acid substitution per 100 amino acids. In overall, if you compare, if you distinguish PAM and BLOSSUM, you can able to find this information, how the PAM matrices are distinguished with BLOSSUM. Okay, more about this you can find from some internet sources. Okay, now let me go to the BLAST tool. What is BLAST? And what are the types of BLAST? Okay, and how to work with the BLAST. So BLAST is a common tool used for pairwise sequence alignment. It is present in almost all the public domain databases. Like it is present in NCBI, it is present in UniProt, it is present in EMBL, it is present in DDBJ. Whatever databases I have mentioned, in all the databases you find this as one of the tool present to check for pairwise sequence alignment. Okay, so what is BLAST actually? The BLAST was described by Lippmann, isn't it? So the BLAST is actually a basic local alignment search tool. Okay, let us go to the website and discuss more about this. So if you go to this website, okay, so defaultly many people try to use the NCBI BLAST. Okay, so if you go to this URL blast.ncbi.nlm.nh.gov, you can also get the types of BLAST versions used. Okay, so if you see the BLAST, okay, the recent version is 2.5.15.0, and the versions, the types are nucleotide BLAST, in which you are using an input of nucleotide query, and you are checking with a database of nucleotide sequence. Okay, and in plastics, you use a query like translated nucleotide. You know what is the difference between nucleotide and translated nucleotide? The translated nucleotide here, I mean, you have to provide only mRNA as the input. So when you're searching the mRNA, when you're searching the sequence, you have to feed only mRNA. You can't give DNA as an input. If you give a DNA as an input and if you search for the blast X, it prompt for an error. It mention you that you're not giving a correct sequence. So only the mRNA alone you have to give as an input. And whatever you're going to try is you're going to search a relevant protein, okay, in the protein database. mRNA is search in the protein database and you get that kind of a protein or related proteins. And in T place 10, use a query as a protein sequence query. And what you will be searching, you are checking with the mRNA database. So you get a kind of an relevant mRNA for your protein. That is T blast10. Input here, you have to provide the protein. I told you how to take a protein, the query sequence, how to take a DNA sequence, how to get a mRNA sequence. Many know how to take a protein and nucleotide from the NCBI website. How to get an mRNA sequence from the NCBI? So for this, better go for NCBI, that is ncbi.nlm.nih.gov. You can still use the nucleotide as a database. Okay, and you have to give any protein name. Now I'm going to give like insulin. Okay, type an insulin. You get both linear DNA, circular DNA. Also, we get mRNA. Okay, now I got here something like mRNA. If you get an mRNA, okay, to work with the blast, what mRNA query is BLASTX, isn't it? This is a mRNA query and it searches with the protein. So now we are going to try 1D with BLASTX. If I try with T-BLAST10, it won't work because the query is a protein here. Can we try this? First, we try with BLASTX. Okay, now I'm going to take one mRNA. So first, initially, I take the FASTA format of a particular species mRNA. Now I'm going to check with run blast. Here we now have an option. Whatever FASTA format you get, you are going to run blast here by clicking this option. If you click here, okay, one thing what we can execute is only the blast X. The other thing, which is T blast and it won't support. Can we try this? I click T blast in. Okay, but the same accession number is there. And now you're going to try with this. Show results in new window. Click blast. See, failed to read the blast query. If you use mRNA, you can use only the selected options. Wherever you find mRNA as an input, only that you can use it. So you can't able to use it in T-BLAST10 as I previously mentioned, but you can try with the plastics. So T-BLAST10 is not in use. Okay, isn't it? Now I close the window. I just selected plastics now. So after selecting plastics, show results in new window, blast. See, it's executing. The program is running because we have given exactly like how we have to give. We have to give only mRNA as an input. Yes, we have provided, and we are searching mRNA with protein sequence database. Protein sequence, and see the what is a protein sequence database we have provided? Non-redundant protein sequence. The database what we search in default is non-redundant protein sequence. It is the NR is the collection of REF SAQ protein, Swiss protein, and PDB. So if you want to check your related protein, okay, through NR, the NR will take care of these three databases. If you want to check only, if you want to get only 3D structure of a protein, then you have to select this database. If you select this database, you get only 3D structure of proteins as the output. But instead, if you want to get even Swiss prot information, even if you want to get REF SAQ protein information, you can select NR, which is present in default. Now check the result. We got the result now from the NCBI blast. So whatever we get from the NCBI blast, even you can download in various formats. If you click this download all, the formats what we can see is text, XML, ASN.1, JSON, okay, text, CSV, XML. So many options are there, including SAM. Okay, it's a common use for next generation sequencing. Okay, these are some of the outputs. Okay, we can find from the blast. So this is a description kind of an output. In this description, we can able to see the scores like max score, total score, query coverage, e-value, percentage of identity, accession length. So whatever we find, okay. So if you want to see this, okay, what is the exact thing? We already searched for the Octodon dangus. So we can't able to go with this. We can go with the next species. We got something like Fucomys tamarinis. Now I'm going to click this accession. So what exactly we are getting now? We are getting, okay, Fucomys tamarinensis protein. I told you now, you get only the, if you search with the protein sequence database, you get only protein as the output. Got it? So we tried with the octo species and we got like Foucault species protein. But whatever we tried with octo is the mRNA sequence. But whatever output we got is the protein sequence. Okay. So if you want to get the protein sequence, you get like this. If you want to get a kind of an alignment, okay, we can also go for this alignments. Okay, the first alignment for the octodon compared with the octodon is yeah, that is not significant. Octodon comparing with Fucomys. Okay, the query is octodon and the subject is Fucomys. If you compare, you get matches, mismatches, and gaps. But instead of mentioning like matches, mismatches, and gaps, in BLAST, you get only identities, positives, gaps. Generally, for calculation of alignment score, we use terminologies like matches, mismatches, gap. But in BLAST, what you are getting in an output, you are getting identity, positive, and gap. Then what is the difference? How we can link with the matches and mismatches? Of course, gaps are gaps, no doubt in that. How about identities and positives? You see this, identities are only matches. Positives are the sum of matches and mismatches. Positives are the sum of matches and mismatches. Gaps are only the gaps in the system. Okay. Likewise, you can able to see the matches, mismatches, and gaps. Okay. So whenever you find only 77 matches, if you check with the other isoform, but you got positives 89, it means you have 12 mismatches. 89 minus 77 is 12 mismatches, and gaps are 3 number. Okay, this is how we can find the output in the alignment format of BLAST. If you compare one sequence with the another sequence, and if you see the graphic summary, this is a third kind of an output. So the first thing, octo versus octo, you get a red color. Why we got a red color? Why we got a red color? Because according to the alignment scores, if it is greater than or equal to 200, okay, then we get this kind of a red color. If you get a less score, based on the alignment score, you get this color. So almost all the comparison, the first 50 sequences have the pink color. And if you go for the next 50, you may get a blue, the green color, then blue color like that. Okay, the most similar are shown at the top, and it progressively go to the bottom. Okay, so the dissimilar sequences, like in ascending order, it will be sorted. And if you want to see this Octo versus Foucault, this is the second sequence, isn't it? You have to click the second pink line. If I click this, you can get this. And if you want to get the alignment, click here. So whatever we have seen initially, the second one, comparing Octo versus Foucault, that we can see here. And of course, this is the third one, third best aligned. And not only that, that are the results. Okay, we also get the few more results. I told you we have max score, total score, query coverage. What is query coverage? Query coverage is like how many characters in the sequence of octo is matching with the Foucault sequence. We got 74% kind of a similarities of the query. The characters in the query is matching with the database. That is query coverage. Then what is max score, total score, and e-value? This we can see in works. So just provide me 2 minutes to complete the blast. Okay, in the blast, we we can also try with the UniProt. Now I'm just trying with the NCBA. One can also try with the UniProt. Okay, and tomorrow I will show you once again how to work with the UniProt. Okay, so you get an output like this. Okay, like how we got from the NCBA, and how to try it in the blast. You should simply give an input sequence. Either give the FASTA format, or you can give the, if it is an NCBA, you give the NCBA accession number or GeneInfo ID. If it is a UniProt, give UniProt ID or FASTA format. Okay. So that related to database, you have to give that kind of an accession identifier, the identifiers, database identifiers, and you have to simply click blast. If it is a nucleotide, defaultly we use NR. NR is the default database. And if it is a protein, no, sorry. If it is a nucleotide, we use NR/NT. Uh, for a nucleotide, NR/NT. For a protein, we use just NR. Okay, for protein NR, for nucleotide NR/NT. And if you see the blast, we have 5 versions of blast. I told you translated nucleic acids we have tried with blastex, isn't it? You can also try with T-blastex, which uses translated nucleic acids, but you blast with translated nucleic acids. So in the previous case, what we tried, we got the protein as an output. But if you try with the T-blastex, you won't get protein sequences, but you get mRNA as an output. mRNA query, if you try with the mRNA database, for this you can go for T-blastex. This is the program we use. Got it? So these are the 5 versions of BLAST. And of course, one can able to know from this slide, okay, what exactly you have to give as an input and what is the output you'll be retrieving from that. So, and this is a graphical kind of a representation. There are types of BLAST, and BLAST algorithm we see on tomorrow, but we can see on how this BLAST output is searched. And these are some of the outputs. What is the bit score and E value? That alone we can complete it now. E value is calculated from the formula K mn e power minus lambda S, where K is a background probability and lambda is also background probability, whereas M and N are the test and database sequences length, whereas S denotes the raw score. The raw score is what? The alignment score, what you calculate in the Needleman ones, or what if you are going for the local alignment, what you calculate in the Smith-Waterman. So whatever sequences you are comparing in the BLAST sequence, it uses, if you have 300 million protein sequences in NCBA, and if you're using an unknown sequence, and if you use a BLAST tool, your sequence is going to search with all the 300 million protein sequences. Okay, through that matrix method. If it is a 110 amino acid, 110 amino acid is going to check with all the other amino acids of a protein. And for all this, of course, defaultly in the computation, the matrix is constructed, and the score, whatever score you get for each and every sequence, which we call it as an alignment score, okay, which is also using an incorporation of amino acid substitution scoring matrix. Even the PAM and BLOSSUM comes into play. So not only the alignment score is calculated through the matrix method, but also amino acid substitution scoring matrix values are also will be included if there is any mutation. Now, obviously, you get whatever you get is the raw score. That raw score, you keep it here for calculation of E value. Got it? So see the complications of searching a similar sequence in BLAST. Our computational power is more, okay, especially the I mean, not ours, the BLAST server computational power. Everything they have in the workstation RAM, so we are getting an output instantly. Okay, they have good storage space and a good processing speed, workstation RAM. And there is also a bit score. S bar is calculated as lambda AS minus ln k by ln2, where A stands for the raw score and K stands for the background probability. And if you want to find the significance of the blast and how the blast algorithm works, all these things we see in the next session, along with the PSA blast and how the PHA blast works, and also how to distinguish blast and FASTA. This will be seen in the next session on tomorrow. Okay, so I have completed my session at present. Any doubts you can ask me. What does is meant by non-redundant protein sequence and what is REF sequence? And could you please also explain shotgun protein or something? You don't bring that shotgun protein at present. Non-redundant protein sequences. So you don't have a single protein sequence for a insulin protein. If you search for an insulin protein in NCBA, in UniProt you have only one entry. For a ligand, you have only one entry. You won't find this entry somewhere in the bottom. It might be a different proteins. But whereas in NCBA, if you search for a particular human insulin, you get multiple entries. Okay, the multiple entries is what we generally term it as non-redundant protein sequence. REF is a protein references. The insulin protein of human can also be linked with chimpanzee protein or even if the suscrofa, pig protein. Okay, for if some proteins are more closely related, then they are kept in clusters, protein clusters. So it also searches that, and also PDB, I think you know that is a 3D structure. Okay, so this is the information. Don't confuse a lot. Okay, with the shotgun sequencing. Okay, it's actually a kind of an after the sequencing you go for the assembly time. Okay, so thank you for your